If you find a path with no obstacles, it probably doesn't lead anywhere. Frank A. Clark. Hello Shishas, welcome to my lecture 3 on cell biology for ninth class. As we are looking at various properties of cells, even going to continue with one of the important topic in understanding the cells is understanding various regions of cells. So instead of calling them as regions, I prefer calling them as zones. Different zones of cells are classified into three major groups. One is called as cell boundaries, another one is the nucleus, and then the cytoplasm. Today, in this lecture, we're going to cover about the cell boundaries and the nucleus. The cell zone 1, the cell boundaries in two living forms like in plants and animals are quite different. When you look at the plant cell, they have a specific structure, additional structure we can say outside of the cell membrane as a cell wall, and the animal cells have plasma membrane alone. So we'll begin with cell wall and its properties, and we'll continue to understand the common cell boundary plasma membrane soon. First, plant's cell wall. The term wall itself says it's quite rigid and strong structure. Whenever you see the plant cell in a microscope, you can see it will have a thick wall, mostly in green color. It's made up of dead cell wall and it is semi-permeable in nature. The structure of cell wall, usually in case of plants, it is cellulose. But similar structure is also seen in fungi, where it's made up of chitin, and in bacteria and blue-green algae, mostly it's made up of peptidoglycan. So the peptidoglycan is one of the strongest structure where our body cells cannot penetrate through inside to kill them. So that's why bacteria are stronger when they enter inside any human host. Alright, so the cell wall under the microscope, it shows four various structures. The middle lamellae are the layers between the adjacent cells, and then the primary cell wall, secondary wall, and the tertiary wall. Let us have a look at the diagrams. Right, this one. Okay, what happened to this? Okay, you can see here how the cell wall appears under the microscope. The primary wall, the outermost one, and then the middle lamella, you can see the gaps are nothing but the middle lamellae here, and then the secondary is the innermost, and then the tertiary is most innermost. You can understand here, so which is the dissected one. The tertiary wall is almost inside, then secondary here, and then primary here. The gaps are middle lamellae. This strongest structure is helping the plant to grow uncontrollable. And if the cell wall doesn't permit the communication between the adjacent cells, it always have more chances of getting some unwanted substances. That's why the plants have certain connectivities with the adjacent cells, mostly considered as the plasmodesmata. The cytoplasmic bridges are gaps between the adjacent cells where they get communicated with the neighboring cells. And now we look at the functions of cell wall. As I told you, it is a rigid structure. Obviously, it protects the inner contents of protoplasm, and it gives specific shape and size, and it also plays a major role in mechanical support of cell. As it is a semi-permeable membrane, it also helps in transportation of water and mineral substances. And now we look at the common membrane of both plant and animal cells. That is nothing but the cell membrane. The cell membrane is one of the structure that has various components. We can say it's discovered through some techniques that it has three major components. So that is proteins, carbohydrates, and then fats. So we should supposed to organize. The scientists found difficult how these are organized or arranged inside the membrane. And this magical boundary is really a typical one to understand. When you say something is completely allowing something, so it is easy to understand, but it is technically allowing few substances by choosing them in a proper way. So that's why the plasma membrane is not good and is not being understand in those days very easily. So you can see some of the historical events there. The term was given by Nageli. It's outermost layer of the animal cell wall, and it is selectively permeable and one of the living membranes. So this is where whenever you say something like life, living, it's difficult to define and to understand. That's why this living membrane was not explained, not discovered at a time. So the models come up to define or to elaborate how the three molecules are arranged in a specific manner. So first model we're going to learn or to understand is the lamella model or the sandwich model, which is given by Danieli and Davison in the year 1935. Let's look at the structure of it. If you see here, the Danieli Davison model, that is sandwich model, they have clearly compared cleverly compared with the bread and the innermost, I mean the middle portions like in sandwich. That's why it's called as the sandwich model. So first is the outermost, the greenish things you can see here are nothing but the proteins, and then you can see the fats. These nothing but the lipid layer, the bilipid or phospholipid layer, and also you can see you try to understand these structures in the middle portion soon. And this model of cell membrane is failed. The reason is the proteins are poor. They have poor capacity to pass over the substances like glucose and fats. So this model did not give proper explanation how these proteins will allow the substances through it, and if you observe the structure of the proteins, they are very difficult, uh, and hence it may not be easily allowing the substances. So this model got failed. It's not accepted by the scientific community in those days, and then we have to move to the next model. So next model is nothing but, okay, I'm sorry, there's something wrong. The next model is the unit membrane model. So the unit membrane model is given by the Robertson in the year 1959. So let's have a look at how this try to explain the model or the structure of the cell membrane. If you look here, this was Robertson's model. So he tried to tell that the proteins are arranged as a unit membrane. You can see the lipid bilayer clearly. The common feature. You can see something. These red heads are nothing but the hydrophilic ones. So when you say hydrophilic, so the carbohydrates are hydrophilic ones. They can be able to get solved in water, and the other regions are called as hydrophobic. So when you say hydrophobic, these nothing but the fats. You cannot mix oil with water. It's difficult. It won't get diluted. But whereas hydrophilic ones, they can be easily diluted. Hydrophilic ones are generally the carbohydrates, CHO, and then the hydrophobic ones are fats. So these are the lipid bilayer is arranged. But still, the Robertson model just replaced the sandwich with the unit membrane. There is no new thing in this, and this also got a failure among the people. So obviously we have to look for another model which clearly describes the model of plasma membrane. So here it comes, the successful model, the exact model how the cell membrane is arranged. So this model is called as the fluid mosaic model. It is given by the scientist Singer and Nicholson in the year 1973. According to them, they have the bilipid layer like in the other two models, and they clearly told that there are two constituents. One of them is hydrophilic one, Another one is the hydrophobic ones, and the hydrophobic ones are buried in the membrane. You can see when you say buried in the membrane, these are going to be they are getting rid of the hydrophilic ones. Mostly the cell requires the hydrophilic ones, so that's why these are submerged inside. And then it has explained about proteins also very clearly. So they found that there are two types of proteins: extrinsic proteins and intrinsic protein. That's what generally the mosaic model, a mosaic stone, refers to the submerged stones. They compared these proteins as submerged stones in between the bilipid layer. So extrinsic proteins, mostly the outer and inner surface, and this is soluble and unstable. And when you say intrinsic, they're closely embedded inside the protein, extending from even sometimes outside as well. And even you can see some specific proteins that are elongated, that are being present inside as well as outside of the membrane. So these are called as glycophorins or phorins. Alright, so let us look at the model of if you look here, there is a model of, so you can see here, so how the outer phase and inner phase. The hydrophobic ones and hydrophilic ones are arranged like this. The head structures nothing but hydrophilic ones are polar, and next you can see the hydrophilic ones. Whenever you say these are phospholipids, all these molecules have the phosphates, because phosphate is one of the important medium to transport the substances. Alright, so next you can see the sugar side chain like glycoporin, and also you can see the integral protein. So this was the key explanation by this model. The proteins are submerged randomly over the membrane, so where these specific places will be allowed to pass through few substances. And even you can see here, these H2O plus, sorry, this refers to the hydrophilic ones, and H2O minus, that is the fat composition. Alright, so let us look at how this membrane or this magical boundary can allow various substances. And now we're going to look at the functions of plasma membrane. Then we try to understand how does this magical membrane can allow the substances in a form. So first, the functions of plasma membrane, obviously it tries to provide the definite shape to the cell, and next they help in important processes like nerve impulses, and also the recognition takes place over the plasma membrane. So it has a lot of function. For example, the macrophages, they always detect the dead RBC cells. Generally each and every RBC cell that is produced by bone marrow, they have around 120 days of lifetime. When they die, they will be moving in the blood, so which is a useless thing. So the macrophages are going to detect them and they remove and transfer to the liver. Alright, so this way the plasma membrane has a lot of functions. Next we'll try to look at how the magical boundary is going to allow the substances through it. In other terms, it is transport across the cell membrane. The cell membrane, for understanding this, even you have to know a bit about few chemical processes. One is called as diffusion and osmosis, and we have classified the transportation across the membranes into two major types. One is called as active transport, even called as biological process in your book, and it is energy consuming or energy utilizing process. Next is the passive transport, which is the physical process and energy is not at all is utilized in this. Active transport is nothing but a kind of loss for the cell, because it should not supposed to waste its energy for transporting something. And passive transport is one of the uh, most seen process in the cells. So first, these things a little bit connected to the chemistry part. Diffusion is where the movement of molecules from high concentration to low concentration, and if there are specific carriers of the substances, then it is called as facilitated diffusion. So here the carriers are mostly the proteins, the protein channels, like we seen earlier. Next important chemical process is osmosis. The osmosis is where the transport occurs from high concentration to lower concentration, but there will be presence of the semi-permeable membrane. And again, osmosis can be defined based on concentrations, so we call them as tonicity, the osmotic gradient. We have three types of tonicities. One is called as isotonicity. Iso, you know that the similar concentration all over, and then hyperconcentration. Hyper refers to more, so hypertension, hyper, all of these are very close terms. So mostly in sciences, whenever there is a term called hyper, that refers to more, and whenever you say hypo, it has less concentration or less tonicity. This way the tonicity will be described. And now we'll try to look at the osmosis. If osmosis is being used to get the substances inside, it is called as endosmosis. The prefix endo refers to in, get in, and exo refers to out, something that can be ejected through osmosis. It's nothing but exit of substances, and mostly if the substances move outside of the cell, it's going to cause the plasmolysis. Alright, so let us look at next thing. This I try to give you what happens to the RBC cell when you expose to various tonicity levels. So first is let us expose to the isotonic. So when you expose it to isotonic solution, there won't be any osmosis and there won't be any change of the cell. And when you expose it to hypotonic solution, the RBC cell ruptures, or you can see it just breaks down. The reason is all the fluid around it, it's going to enter inside. So when it enters, it gradually bulges its size and blast. Next is hypotonic. So in hypotonic solution, what happens is the RBC cell become flaccid. So flaccid in the sense it's going to become very thin. All the substances inside the RBC will come out. Hence you can compare. It just become thinner and then there won't be anything. Even it is called as plasmolysis. Now we'll look at the process of transportation. So this process, endocytosis, again you can see how the substances will be taken inside the cell. It is called as endocytosis, and here you can see just imagine the cell is getting ready to have its dinner. It's waiting to have something to eat and something to drink. If the cell is preparing to eat something, then it is called as, sorry, let's say the drinking, it's called as phagocytosis. So it refers to ingestion of substances. So the specific body that helps in this is called as spinosome. And here we have another way. So it wants to eat something. So then it is called as phagocytosis. The pino, phago, or Latin terms. The phagocytosis refers to the cellular eating. The cellular eating is where ingestion of substances, mostly solid complexes, through the body called the phagosome. Alright, so these are the two methods of bulk transport inside the cell. That's why it is called as endo plus cytosis. And next we have one more method called exocytosis. How does the substances will come outside of the cell? So here you can see it has various names: exocytosis, ephagi, cellular vomiting. Here the egestion or getting rid of the substances. Alright, now we'll try to look at an illustration which helps us to understand in a better way. This is the one. Okay, you can see here the phagocytosis. The phagocytosis is where the cellular eating part. You can see the plasma membrane is making folds outside of the membrane. So this is a linear membrane, and you can see clearly how the membrane is produced outside of the cell membrane. So this way the solid particle enters inside the cell. And next part is the phagocytosis. When you see the phagocytosis, outside you can see a few molecules are waiting, and the membrane is forming the opening inside, or the sac is being produced inside, and then the membrane will come close and it just closed. Okay, you can see how the molecule is taken inside the vesicle, and then vesicle moves towards the cytoplasm. And the receptor mediated endocytosis, so mostly this type of endocytosis seen for the neurotransmitters, and you can see the receptors being added with a few signal based particles, and then it's going to be, all this region is going to form a vesicle and new membrane is going to cover it up. Okay, so this way also the endocytosis may happen. But remember, phagocytosis, the cellular drinking, and phagocytosis, the cellular eating, are important processes. Now we'll try to look at the only method of the exocytosis, or removal of substances from the cell. So here you can see how the substances can be moving outside. First, if you look at here, the plasma membrane is going to attach towards the membrane. You can see the Golgi apparatus is actively making a vesicle, and then this vesicle is enclosed with the newly synthesized proteins, and also it has tagged proteins here, and this going to get in touch with the membrane, and the particles will move away, and then the same things will remain at the membranes. Okay, and this is quite difficult to understand, but whereas here this one is commonly explained. So some of the molecules like proteins, the Golgi apparatus is going to form the vesicle, they move towards the membrane, and you can see the integral proteins here. So the signal proteins are present here. As soon as it come close, and it gradually going to merge with the cell membrane, and you can see the particles are going outside. So this way exocytosis happens inside the cell. Remember, exocytosis may cost cell. So generally exocytosis requires the ATP molecule. The entry is free, but exit always requires this currency called ATP. And let me explain you one more thing here, the facilitated diffusion, one of the illustration I collected for you. So the facilitated diffusion requires some carrier proteins. If you remember, the carrier proteins are present in between the membranes, and as soon as some substances like glucose, when they come here, and you can see they gradually come inside. Okay, so this is a type of facilitated diffusion, very often seen in case of the glucose and then few minerals. Let's go back to the other part. These all about the cell zone 1, and now we'll try to look at the cell zone 2. The cell zone 2, as we have seen, is the nucleus. The nucleus is also called as the center of cell functions. All the functions of cells are controlled by the nucleus, except two structures, but even they are under the control of nucleus. First time the nucleus was discovered by Robert Brown in the orchid cells. So orchids, some of the plants that are grown for decorative purpose, and there is a common rule that every cell should supposed to have a single nucleus. Exceptionally, there are few things which lack this, which are away from this rule. Example is matured RBCs in animals or mammals, and then you can also see the sieve tubes where they lack the nucleus for a good purpose, of course. Again, this cannot be considered as a common rule in case of all mammals. There are RBCs in the blood of camel and also the llama. These organisms have the RBCs. And now we look at the structure of nucleus. So the nucleus is one of the central structure of the cell. It has four different regions again. First is called as nuclear membrane, and then the nucleoplasm, chromatin net, nucleolus has many names, little nucleus or ribosome factory. If you look at the nuclear membrane, this separates the nucleus from the other portions of the cell. So nuclear membrane is connected with endoplasmic reticulum mostly, and then you can see there will be small pores or holes that allow the substances produced by the nucleus to move outside, or to get the substances from cytoplasm to inside. And also you can see a lot of proteins like RNA, DNA complexes which will be exported to the cytoplasm. So remember, the nuclear membrane is a protective shield. At the dangers, the nucleus will be preserved without undergoing any immediate changes. Next structure we're going to learn is the nucleoplasm, the similar cytoplasm found inside the enclosed nuclear membrane. So you can see the nucleoplasm will have a rich chemical substances like nucleotides. The nucleotides and nucleosides are quite common term. Only the term S is a different. So nucleotide and nucleoside. So here in brackets I try to clarify you what is meant by nucleotide. When you say nucleotide, it will have a sugar and then nitrogen base. And uh, when you say it is joined with the phosphates, then it will have the complete thing. Okay, so till here you can say it's nucleoside, and when it joins with the phosphate, it becomes nucleotide. The equation that follows for the genes or the fragment of DNA is like this. DNA will have the specific bases which are paired to specific bases. They follow this rule in nature. So A always pairs with T, C always pairs with G. And also the pattern of RNA also is very specific. Instead of T or thymine, it's going to have the uracil here. So A always pairs with U and C always pairs with G. Remembering the pairing with C and G is quite easy, but you have to be sure that DNA is getting joined, adenine with thymine and RNA with uracil. And now let me show you a small illustration to understand this, and mostly you'll get a lot of questions based on the pairing of the bases. So some basics here. The nucleotide. So first you can see the nucleoside first, the nitrogenous bases. You can see the bases, and also we have two types of sugars, the ribose sugar and the deoxyribose sugar. So see, whenever you say ribose, you can see OH group. When you say deoxyribose, it is H group. That's why the name itself, the ribose and deoxyribose, it doesn't have oxygen on it. So simple in terms of chemistry. And then you can see two types of nitrogenous bases. The purines are 2, adenine and guanine. The pyrimidines are 3, thymine, cytosine and uracil. So cytosine is a common structure for 3, and uracil is only seen in case of RNA and thymine in case of DNA alone. Next we can see the phosphate groups. So the phosphate groups are 3. If it's 1 mono, di, 3 tri. Even this nucleotides are not only helpful in passing the genetic information. Interestingly, I think you know the full form of this adenosine triphosphate. See, the triphosphate means it has this structure whole, and also you can see adenine, so a type of polymer of adenosine. Do you know this thing is really money of the cells? They exchange this as a form of energy. So adenosine triphosphate, like this, there are many interesting facts that are lying behind the chemical structure of the cells. Now let me illustrate you one of the concepts of how the cells are arranged. If you see here the DNA sequence, first we'll understand DNA sequence. So C pairs with G with triple bond, and A pairs with T with double bond. So this is being a common rule. And even you can see the 5 prime 3 prime, the double, these are called as double stranded DNA. One side it will be from 5 to 3 and 3 prime, and the other side it will be 3 prime to 5 prime. Okay, that's why it is called as antisense. Okay, and when you see this exactly DNA sequence wanted to get converted into RNA, you can see some changes as you followed the universal rule that A always pairs with U in case of the RNA. You can see you don't see any T here, right? And also the C and G are quite common, but A and T are not. So you can always see A pairs with U. So be clear with these questions. So the DNA sequence, if it is double stranded, it will always have the 2 strands parallel to each one. Alright, so these about the DNA and its structures. And next we learn about the chromatin net. Even sometimes you can see the same thing being considered as the chromosome. So chromatin itself nothing but long thread-like fibers that are found in the nucleoplasm, and also it is mainly formed of the DNA and histone proteins. DNA you know already, and histone refers to the specific protein that packs the chromatin net. So you can't expect the DNA to roam in the nucleoplasm so easily. It should be packed in a proper way so that it will be safe in the rest period. And when you say something like chromosome, they always appear during the time of cell division. Okay, you can see the condensed form, and then the important structures like this. You can see the region where they joined is called as centromere, and other regions you can see the chromatid threads. Okay, like this, they grow like this, and they join at the centromere exactly. So next structure, final structure is nucleolus. So the nucleolus was discovered by Fontana. The term was given by Baumann. Nucleolus is generally darkest structure in the nucleus, and it is a membraneless structure. It is quite round, and also it is attached. It's usually attached to the chromatin. Alright, the main function of this is to produce the ribosomes. There's no other function. A nucleoli is absent in few places where they do not require to produce any ribosomes. For example, the sperm cells, where it doesn't need to produce any proteins. What is the function? It has exact chromosomal number that requires. And next is the muscle cell. So these structures lack the nucleolus inside the nucleus. Next, finally, we reach the end of this, the functions of nucleus. The nucleus carries the genetic information of the species, and then they also help in transmitting the genetic information from one to another one. And the division of nucleus is a primary step of the cell division. And also you can see we are so varied. We have a lot of variations. You can't expect you to be 100% exact like your parents. You have some variations from your parents, and they are varied from their parents. So all these variations are due to the magical matching of the genes which are found inside us. So that's it from me today, Shishas. Let me meet you with the next lecture. See you.